Good morning. My name is Alfredo Napoli. I'm a researcher in a French center called CIRAD, which is located in Montpellier. And I'm very happy to present today some results we have obtained on the biochar production and characterization in this summer camp of the AGRIMA project. So my presentation is called Physico-Chemical Characteristics on Biochar for Remediation and Sustainable Economy, Main Challenge on Pyrolysis Process Development, and study case with the European Acronym project. So the first point is thanks a lot for this invitation of the AGRIMA project and the opportunity I have to present some results, which is of course connected to the waste management for sustainable bioeconomy. And the idea of my presentation is to transfer for you some idea about the methodology to study one case, which is biochar production and use, and also your strategy and challenges. So here in this presentation, in this slide, some indication about the main objective of the AGRIMA project, which is mainly focused on the transfer some pedagogic methods for on the valorization of agro-food waste and how to connect all participants, all activities in terms of research, in terms of industrial companies and the society, and also to transfer and to involve some citizen community on these problematics. So the main partners of the AGRIMA project are represented in this slide, so which is mainly a European project with different countries from Europe. So to start my presentation, before starting, I would like to present quickly what is CIRAD. So CIRAD is a French research center which is mainly focused on the international cooperation with tropical countries and dedicated to the agronomic research with one finality, which is to be involved in the development of these countries. CIRAD is mainly research center located in Montpellier in France, in Montpellier where there is a technical staff, and we are more or less 1,750 people, more than 1,200 scientists, with a annual budget of 240 million euros per year. And we have 3 departments, which are the BIOS department, PERSIS department, where is my research unit, and the ES department. The first one is treat of the biologic aspect of the agronomic thematic. The second one on the processes to produce and convert and transform food and biologic biomass into end product. And the third department is focused on the environment and society studies. So in this presentation, I have been invited to present activities on biochar, and my question and how to present it was, okay, what are the kind of challenge I would like to present to you, and what is the strategy we have developed at CIRAD in order to work on this thematic and try to solve these challenges. So for that, thinking about this idea, I think that one relevant presentation may be about the activity we are in progress today with a European project called Acronym, where the objective is to capture and reuse biogenic gases for negative emission and sustainable biofuels. And this project is about duration of 45 months with 13 partners from European Union. You can see on the map the different partners and countries involved. And it's a European project which is dedicated to the thematic sustainable, secure and competitive energy supply. Okay. So what are the objectives of the project is mainly to develop technologies for biofuel production and the utilization of biogenic effluent gases. So the idea is to work on new technologies and at a scale level, which is from a laboratory scale with a TRL 2 or 3 and to pass up to a TRL 5, which is more or less a small pilot plant. And there are 5 technologies which are which have been selected for the pertinence relevant for this objective. One of them is about the biogenic carbon storage through biochar production and involve a pyrolysis process. In this project, one part is to analyze also the technical, the economic, and the environmental assessment of the technology in order to check if it's good or not to be developed in the future at a larger scale. So just one thing is about the prototype about pyrolysis and the biochar. So we have in the picture here the idea of the project, which is to use and develop a pyrolysis pilot plant, which can be used with some raw material, which are here represented. It can be a biomass, it may be agro waste or solid digested, and through conversion, a thermal conversion, we can produce 3 main co-products. One of them is a solid product, which is called biochar, and which is mainly a concentration of carbon, solid carbon, like charcoal. The second and third co-product are the pyrolysis gas and pyrolysis oil, light gas, which called light gases, and these 2 co-products will be used to produce energy. So the idea is to convert biomass into a concentrate carbon solid phase biochar, which can be used in future in agronomic for both improve the performance of some cultures, some crops, and also to improve the carbon sequestration in soil. And the other part is to use a fraction of the biomass into co-product, gaseous co-product to be used in energy. And the idea is to produce in one part energy for different end user, and the other part is to use the solid fraction to assure both the agronomic performance and the productivity and carbon sequestration. And the idea is to have a carbon balance of both applications, which may be negative as indicated in the project. So firstly, the main question is about the challenges. So in this picture, I would like to present to you the 3 main challenges I think is we have to treat in the project. So the first one is, so you have here the picture. In this picture, you can see the biomass, you can see the pyrolysis process, which is a thermal treatment in the range of 400 degrees C up to 800 degrees C, 1000 degrees C, and we produce the solid and the co-product here. So the first challenge is to achieve at a technology which allow to convert into solid and co-product, and the first one is to use a fraction of the co-products for energy inside the reactor. So that means that this first point is to ensure that the energy produced here, a fraction of the energy produced here through the coproduct, can be used directly in the pyrolysis process in order to maintain the process in the range of 400 degrees C or 1000 degrees C, up to 1000 degrees C. So the first one is how to use the first coproduct, part of the energy in the coproduct inside the reactor to assure the energetical autonomy of the process. Well, this is one point. The second point in relation with the second challenge in relation with the biochar production and bioenergy is to assure, to optimize the production of the coproduct for the energy final use of the energy and the biochar production here for the agronomic valorization and the carbon sequestration. So this is the second challenge, is how to optimize the production of the biochar and production of the coproduct for energy, always maintaining the autonomy, the energetical autonomy of the process. And the third challenge at the end is about the characteristic of the properties of the biochar in relation with the application, the final application, which is mainly in relation with the agriculture and carbon sequestration. Otherwise, in another word, how we can, with the three challenges, how we can produce technology, we can allow us to use a fraction of the energy inside the coproduct to be used inside here for the autonomy, to optimize the production of energy and carbon sequestration, and to control the quality and the property of the biochar in order to be in adequation with the final use in soil, both in terms of agronomic performance and carbon sequestration. So in this slide, we have the challenge, and we would like to work in these three challenges in order to try to solve it, or almost to collect relevant information in order to develop at the end pilot plant and at term industrial level process. So for that, the question is, okay, we are talking about the biochar, which is for agronomic application, but how we can imagine today what is the situation. And for that, I would like to present here another biochar, not biochar, but charcoal use technologies, which are industrial technologies, which are in the world. And so in order to have the idea of what is today the situation in terms of technology, pyrolysis technologies, the best way to evaluate, to assess the level of development of pyrolysis technology is to check with the charcoal production in the world. And here you have picture, or in the world, the different technology we can use for charcoal production, which is similar to biochar production. So you can see here in tropical countries like in Africa, Madagascar, Cuba, for instance, some technologies, rudimentary artisanal technology. Here you have in Brazil some small kiln to produce charcoal for the steel maker production. Here you have also in Brazil, and you can see here the furnace with a big volume. And here you have in Europe some technology with a chimney here in order to collect and burn the co-product to burn it. And here the most developed technology, which is in Brazil, a continuous furnace where you can produce charcoal and collect all the co-product for valorization, as explained in the last picture here. But in this case, it's only with wood feedstock, and with the finality is a charcoal for steel maker and cooking furnace, and not for biochar and soil. So with this situation, in this context, so what are our strategy at the BioWeb Unique Research Center Laboratory for pyrolysis study, taking into account all this context and challenges? So we have decided to work so we have decided to work with a project of Acronym, and the methodology we have selected is to work with a different raw material. So we have decided to work with three main raw materials. The first one is wood chips, because it's a class of biomass. The second one is agricultural residue, a model of agricultural residue, and we have decided to work with rice, agro-residue, so both the rice husk and rice straw. And at the end, we decided to work with another type of feedstock, which is solid digestate from methanization. And the capacity we have is to characterize the raw material. Firstly, of course, the basic analysis, which are the proximate analysis, firstly with the fixed carbon, the volatile matter and ashes content in the biomass, and also the energy content in the biomass, so the high heating value. Of course, we can also analyze the elemental analysis with the carbon, hydrogen, nitrogen, and oxygen compound inside the biomass and the ashes composition. And at the end also the cellulose, hemicellulose, and lignin content we have in each biomass. So the pyrolysis study is based in three levels. We decided to work at three different areas, which are characterized by the quantity we can use and the final information we can collect it. So the first one is the thermogravimetric analysis, where we have developed equipment and we can work with some sample of up to 1 gram maximum, and we can work with the pyrolysis condition at different temperature but also at different pressure and with different carrier gas, nitrogen, CO2 or steam. And all these data, so we measure the evolution of the weight loss during the thermal treatment, pyrolysis thermal treatment, and we can determine the curve of the decomposition of the raw material into biochar and co-product, and then we have the biochar yield with time and temperature. So here you have an example of the curve. So that means that we can define what are the main parameters in terms of temperature, heating rate, the atmosphere of the gas, the pressure, and the influence of each parameter on the evolution of the conversion, thermal conversion. The second level of the methodology is at a laboratory scale with a furnace, a classical furnace, which is here, where we can work up to 10 grams more or less, or 20 grams. It's in relation with the density of the raw material. And then we can work in order to determine the heat and mass balance and the gas composition of the pyrolysis. For that, we have equipment here where we can do the thermal treatment, and after that we have a condenser and a pyrolysis gas analyzer with a chromatography online and continuously, and so we can determine the biochar yield of the final solid we obtain in the furnace. The condenser allow us to condensate all the pyrolysis oil and determine by weight the quantity, and then the bio-oil yield. And with the chromatography, we have the evolution of the main gas compound during the pyrolysis. We have here an example of the curve we can obtain. Of course, after these two studies, we can characterize the biochar produced in terms of pH, in terms of point of zero charge, which are two main information for agronomic application, and also in order to better understand the structure, the textural structure of the biochar, we can work on this, analyze the surface chemistry and surface area. All this information allow us to determine what is called key performance indicator, KPI, and then on the base of this indicator, in terms of just to think about the three main challenges and compromise optimum for the process, we can, through the key performance indicator determine what are the main conditions for a good pyrolysis in terms of energy production and biochar production and performance. And with that, we have a third level, which is at pilot scale, with a prototype we have developed, which is more or less 5 kilograms of biomass treated. And we can produce biochar in different conditions in relation with the optimum defined here in order to do some greenhouse trial at the end. So now I would like to present to you the main information results we have obtained through the Cronus project. So here we have the results of the pyrolysis of the 4 feedstock, which are the rice husk, rice straw in one part, the oak wood, and the digestate. And here you can see the evolution of the weight during the treatment under the nitrogen gas, CO2 gas, or steam gas with a temperature up to 800 degrees C. And you can see that the behavior of the treatment is different in relation with the gas nature, type of gas. And so you can see, for instance, you can see here that with nitrogen, CO2, or steam, we have no difference up to more or less this temperature, which is more or less 750 degrees C. And then in relation with the gas nature, we have a second decomposition, which is called activation. So that in fact we transform biochar in no porous biochar into a porous biochar, like activated carbon. And so you can see here the evolution and the impact of the CO2 or steam as an activation agent to convert it. And there you have the final biochar yield under nitrogen or under CO2 or steam gas carrier. And you can see here the same thing with oak wood. You can see here there is no difference here in this part, or for the digestate, or you can see here the very big difference between pyrolysis under nitrogen or CO2 or steam. So at the end, we can see that the activation appears always after a temperature of 750 degrees C, and that the steam activation is higher than the CO2, you can see here, or perhaps here, or in this case for wood. So you can see that in relation with the biomass use, the feedstock, we have a behavior in terms of pyrolysis and activation and at elevated temperature, which are very, very different. And so we can assume that we have a different product, both in terms of co-product, pyrolysis gas, and pyrolysis oil, and the biochar product. So, just one thing for the students who are present here in this symposium. These results are very nice. It's very nice to see the curves and to analyze, but we have to think about that when we are working with experiments, always you have some trouble, and for that I'm very happy to show you some preliminary results and difficulty we had with this test. Here you can see, for instance, that we work with a digestate, with a rice husk in this case, and the first activity and measurement with steams was very complicated. Why? Because we have developed a specific equipment here, a steam generator system, in order to work with activation with steam, and you can see that we have some trouble in terms of mass gain here, in terms of trouble here, etc. So we work on this part a lot in order to better understand why we have these difficulties and problems, issues, and determine that the methodology we use, the protocol we use with steam has to be quite rigorous, because we have some condensation of water during the reactor, and then it is very important to adapt the protocol in order to avoid this kind of problem. The another point, very interesting, is about the digestat and with team, where we have done work on this activity, and we have seen that here you can see the crucible, and we have seen that with a digestat and under steam activation at elevated temperature, we have some broken here. The work is corrosion appears and some difficulties. So we have decided to work with another. It's steel. In this case, it is steel, and so we have decided to work with another support, which is with platinum, and resolve it. So it was just to show you that experiment is not so easy, and sometimes, the most time, we have trouble, and we have to think about that and try to solve it. Another point is about the pressure. We work, as I told you, we work also with pressure. So here you can see the results of the pyrolysis of the different feedstock under atmospheric pressure and 5 bar pressure. And we can see that the evolution of yield, of biochar yield. The idea for us was to check if the pressure can improve or increase the biochar yield for different theoretical reasons. We assume that, and then we work on this part, and we can see that the pressure, almost 5 bar, we have a small difference in terms of biochar yield, both for the risers and the rise slope, a positive influence, and the Hawkwood, but for the digestat we have a negative influence, and we don't know why. But in any case, we can see that the pressure has a small influence on the positive influence generally on the biochar yield. Okay. Another point which is important, we can see in this part that we have no modification. So that means that the conversion of the biomass into carbon, solid carbon here, there is no effect of the pressure. The pressure is only after 500 degrees C, more or less. So we can assume that the impact of the pressure is mainly focused on the secondary reaction. That means that with the co-product produced after the biochar production, in order to produce more solid carbon inside the biochar matrix. So it is one hypothesis, and we would like to work more on this. The next slide is about the laboratory scale with 10 labs. Here we can see the mass balance. That means that the distribution of the biochar, gas, pyrolysis gas, and biohol produced in the at different temperature for the different biomass and feedstock. So here you can see the distribution in terms of biochar, bio-oil, and the gas for 3 different temperatures, which represent the range of pyrolysis study. We can assume that the pyrolysis starts at 300 degrees C. Below 300 degrees C, we are not working with a pyrolysis process, but more than torrefaction. And up to 800 degrees C, we can assume that we are in the process of pyrolysis. Of course, we can work with a higher temperature, but in terms of technology, in terms of material, etc., it's a little bit more complicated. And if we are focused on the industrial development at the end, we can assume that the range of 300, 800 degrees C is a good range for the study. So you can see here the evolution of the biochar yield with temperature for the risers, the rise throw, digestat, and Hawkwood. So you can see that we have a decrease of biochar yield, but mainly between 300 and 500, and after we have a stabilization, more or less in all the cases. And on the other part, for the bio-oil, we have an increase of the bio-oil yield and the gas, pyrolysis gas yield with the temperature. You can see here that we have more or less a constant evolution between 500 and 800 degrees C of the bio-oil production, more or less, except for the digestate. And so here we increase the gas, so that means that we can assume that the evolution of the gas is in relation with the decrease of the biochar production, and not impacted through the bio-oil, which is here. At the same time, we have the same result, but not for the mass balance, which is here, but now for the energy balance. What's mean energy balance? Energy balance means that we have the energy contained in the biomass, which is here, and we analyze the product, the final product, biochar, bio-oil, and gas, and calculate or measure the energy content in the biochar, bio-oil, and gas, and then we can determine the part of the initial energy contained in the biomass, which is present in the biochar, bio-oil, or gas. And so here you have the distribution of the energy in the different final products. So you can see that in this case, we have 72% of the initial energy of the risers, which is contained in the biochar at a pyrolysis of 300 degrees C, and then you can decrease up to 45%, and you can see that the tendency is the same for all the product, like the mass balance. And you can see that a significant part of the energy of the biomass is contained in the bio-oil, mainly after 500 degrees C and 800 degrees C, and we increase a small part of the energy inside the pyrolysis gas. And we reduce here in the biochar the energy content with a fraction which is more or less between 35 up to 45%. So that means that depending on the temperature, we can have the half of the energy is contained in the biochar, which is a solid, and half of the energy is contained into the co-product, which are the bio-oil and gas. So this information is directly important with the challenges I presented in the last picture here, where we have more or less 50% of the energy here and 50% of the energy here. It's correlated to the carbon content also, we can see after. Okay, the mass balance, the energy balance. Well, so we have a mapping of the distribution of the mass balance and the energy balance in relation with the type of feedstock and the pyrolysis parameters. And now the other question is the characteristic of the biochar. So the first one is the elemental characteristic, and we can see that for the different product that we have 50% in carbon here for the different temperature here. Hydrogen, we have a reduction with the temperature, nitrogen also, and oxygen we can reduce. So that means that with temperature, we increase the concentration of carbon inside the biochar, and we decrease the oxygen and hydrogen with the temperature inside the biochar. Okay. Another point of characterization is the proximate analysis in order to determine what is the moisture content, the ashes, which is very important, and the volatile matter and the fixed carbon. One point which is very important is about the fixed carbon, which mainly can be represented like thermal stable carbon. So that means we can assume that it's a carbon which is very stable inside the soil. So in terms of carbon sequestration, we can focus on this information here in order to check the potential of carbon sequestration. So you can see that with the temperature, always the same thing. Here we have the risers, rice straw, digestate, and lockwood. We can see that we firstly we increase the fixed carbon with temperature. So that means that elevated temperature allow us to obtain a biochar solid carbon product with a very strong links of carbon. So that means in terms of carbon sequestration is good. Another point which is very important is the ashes content, and we can see that in relation with the different type of biomass, we have different content of ashes. For oak wood, we have very low ashes content, when for the risers and the rice straw, we have a very elevated up to 40% of ashes content. So that's part which is very important, particularly in terms of ash composition inside the soil and has a nutrient perhaps for the different plants and soil. One other part is about the digestate. We can see here that the content, the fixed carbon content is low compared to the other type of biomass, and we have a very strong high ashes content. This is normal because digestate, we have to think about the origin of digestate. Don't forget that digestate it's first biological decomposition of biomass in order to produce methane, and so we have a no treated final biomass or low treatment, biological treatment with a digestate, and so that means that we have a small fraction of carbon, but a very big fraction of ashes. So another point is about the characteristic of the biochar in terms of textural properties. Here we have some analysis of the main surface area of the different biochar obtained from the different biomasses. Here you have oak wood, the rice husk, the rice straw, and the digestate biochar when they have been activated through CO2 and steam. And so you can see that we increase the surface area. Here in this case for oak wood we can achieve 650 square meter per gram of product, up to 1000 with steam. And for other feedstock we have a very lower surface area. One thing important is that for the digestate we have no surface area, so we have no so it's a very important information in relation of the behavior of this digestate in future with in the soil. Why surface area is very important for us? It's because all the reaction we have in the soil through the nutrient and the biochar is based on the surface reaction. So that means that if we have a very large surface, we can increase and improve the reaction. Another point about that is the distribution of the pore, and so in this case, as we work with nitrogen and CO2 in order to check the distribution of the pore, and we can see in the future slide, in the next slide, the influence of the steam and nitrogen and CO2 on the pore distribution. So here you can see a picture exactly on the distribution of the pore of the biochar, where firstly we can see that the value here of the pore width is from 0 up to 2 nanometers. So we are in micro porosity system in all cases, both from oak wood, rice husk, and rice straw. And we can see that if we have activation through CO2 or with steams, we have a different distribution. In fact, water allows us to increase the distribution of the pore of the biochar in all cases. Another point which are really important is the pH and the point of zero charge of the biochar. On this picture, you can see for the different feedstock, you can see the pH. This curve is the final pH we obtain of a solution, initial solution with a mixture of biochar in relation with the pH of the initial solution. So we have initial solution. We prepare initial solution at pH 1, 2, up to 12 or 30 here, and we mixed with the biochar, and after 24 hours we can measure the final pH, and we have here the value of the final pH. So this curve allows to determine for us firstly the pH of the biochar. That means that we have an initial solution at pH 7, and we can see the final pH of the solution. But also we can determine when we have a modification of the behavior, surface behavior of the biochar. It's when we have the final pH is equal to the initial pH of the solution. So at this point, the intersection of this curve, which is the pH initial equal pH final, and the curve here, we obtain the point of zero charge. And another point, which is very, very important, it's a behavior here, where we have a buffer behavior of the biochar in this range. It's very, very important for the agronomic aspect, because the buffer activity of biochar or any other product is very important, because we have in agronomy the idea to maintain the pH of the soil between more or less 6 up to 7, 7.5 in terms of pH. So if we have a product with a buffer activity, it's very nice and good for us for the agronomy aspect. And here you can see we have a buffer for the different temperature of pyrolysis, but we have a behavior, different behavior in relation with the feedstock nature. Another point is about the co-product, and here you have, and particularly the gases, and here you have the composition of the gas in terms of dihydrogen, CO, CO2, methane, and the another gases in relation with the temperature of the pyrolysis and for different raw material. And we can see that with temperature we mainly produce CO2, which is the main gas product during pyrolysis. We can decrease it with the temperature, as we can see here. We have CO here, we produce CO. Another point is that with elevated temperature we produce dihydrogen, only with temperature of 800 degrees C. Below, we have not production of the hydrogen in the co-product gas, co-product pyrolysis gas. This is very important for a different application at the end. Okay, so with all this information, we achieve to one step, which is to determine the key performance indicators. In this case, we have decided to use some indicators which are represented here, and mainly is about the bioenergy efficiency, the CO2 emission in relation with the initial biofuel produced, the carbon capture, and also the biomass resource utilization. So we have different indicators, and we can apply it in order to determine for each biomass the different indicator. What is very important in our case, which is biochar and energetical co-product, it's mainly to check what is the carbon capture used during the process, so this indicator, and another one is what is the bioenergy efficiency. That means that the energy content in the final co-product in relation with the energy content of the initial feedstock. These two parameters are the most important parameter for us. Okay, and so we can, in this case, we can check that we have more or less efficiency in terms of capture, mainly with oak wood and rice husk. And here for digestate, we have a lower capacity of carbon capture and sequestration. And another part is about the energy use, where we have a good one here, but you have also here and here, more or less. So the idea is to define what kind of biomass is very relevant for both carbon capture and bioenergy use with the co-product. Okay, so now with this indicators, for us, we have decided to work mainly with rice husk and oak wood, and perhaps at 500 or 800 degrees C, but we decided to work at 800 degrees C. One important reason for that is carbon capture in terms of labile carbon or not labile carbon. I have no time to explain more about this point. So the next point is about pyrolysis device at pilot scale and to work on this activity. So we have developed a pyrolysis unit at the BioWeb research unit with the platform energy we have. We have the opportunity and we are lucky to have an energetic platform where we can develop some pilot of pyrolysis and another type of reactors. So we have a furnace here, which is represented here. We can work at 1000 degrees C with a heating rate of 10 degrees C per minute maximum. And we can work both in nitrogen condition with injection of nitrogen gas or CO2 injection for the activation as we discussed before and studied. So here's some picture of the biomass we have used and the capacity. So we have some crucible here, and we can work at batch mode or continuous mode. It depends on the type of biomass and the capacity of the furnace and the type of biomass. Okay, here you can see we work mainly in batch condition. So here you have some pictures of the rows of the rice straw, rice husk, and the biochar products. Here we have some wood, which is coming from industrial partners, directly from pyrolysis industrial companies. Here you have some pictures about the PID, the system to control the furnace, and some equipment to measure temperature. Here you have the gas flow meters we used. And we have, firstly, we studied also, so you have some basket here in the different part of the reactor, and we studied the influence of the pyrolysis on the heat of each basket. So here you have a picture of the reactor and the software we have developed in order to control all parameters, the temperature, the setup, the flow, the gas flow, the heating rate, etc. And to collect also the data in terms of temperature, profile of temperature inside the reactor. Some picture of the connection outside. So we have the pyrolysis gas which are going out from here to the external combustion chamber here in order to burn it. Here you have the exhaust pipe to transfer the pyrolysis gas from the reactor up to post-combustion chamber here. Now the pyrolysis trial. So we work firstly, one point was to determine the upscaling influence. So that means that we have to compare the biochar yield and characteristic obtained from the pyrolysis system compared to the laboratory scale. And for that, we have done some preliminary tests at batch condition with different temperatures, 3, 5, and 800 degrees C under nitrogen. And after we worked at with the three biomass which are here under nitrogen and CO2, and we can present some results about that. Firstly, the biomass density. Here for each basket we have defined we complete each basket of the pyrolysis unit with biomass, the different biomass, rice straw, rice husk, and oak wood, and measure the weight of each one. And you can see here the weight of each basket with the biomass, and you can see that the density of each straw is very different. We have 100 more or less up to 300 kilograms per cubic meter. So it's very important to take into account this parameter for future development of process. Another point is about the repeatability test of pyrolysis. Here we work at 800 degrees C under nitrogen, and we have here the heat for each basket, which are here. At 800 degrees C with the husk here, biochar. So you have the heat of biochar heat for each basket. So you can see you have a homogeneity here of the result for each basket inside. So that means that we have here the homogeneity of the treatment in this part here, and all are in the phase of 35%. Okay, and now, so we have good repeatability, and the main idea now is to compare with the laboratory with the results obtained at laboratory scale. Here you can see the mass balance here, and you can see that in the same condition we have a 37.8% of biochar at 800 degrees C under nitrogen, which is very similar and close to the biochar heat obtained at pyrolysis pilot scale. So it's a very good result for us. Another now it's another result. It's about the influence of the temperature under nitrogen. So we work at 800 degrees C under nitrogen, and we have done one test under CO2. And so here you can see the biochar heat. Always you can see the homogeneity in the basket, on the 4 baskets. Here, and you can see the evolution of the heat if we compare at 500 and 800 degrees C, and here you have a small difference, 800 under CO2 or nitrogen, where we are on a small part of activation, and we can see also we have no good homogeneity here. One point it's important is that the CO2 is injected by the bottom of the pyrolysis reactor, so perhaps we have activation at the bottom, and after that consume it, and here we have no more activation. And in the same way, we can compare the result at laboratory with the result at laboratory scale, and we can see here we have at laboratory scale at 500 degrees C 32%, which is exactly the same, and here 28, which is more or less the same. So we have also very good results and very close and between the labs and the pilot scale results. And the biochar heat decrease with the temperature, and we have a small effect of the CO2 for the activation. Another point now, it's the results we obtain with the oak wood. And here we have exactly the same type of behavior in terms of results at laboratory scale and pilot scale for the oak wood. Okay. In terms of biochar characteristics, so we can see here about the ashes, we have more or less the same thing, and about in terms of fixed carbon, which is more or less similar, and you can see the same behavior for all. One point is here in this part, we have, well, of course, we have here rice straw ashes content, which are very higher than for oak wood. It was the same at laboratory scale, so there is no problem with that. Okay. And at the end, so we have studied at laboratory scale at different level. We worked at a pilot plant in order to produce some biochar. We have seen that the behavior and characteristics are similar, and now we have the agronomic trial to do. And for that, this activity is on progress, and we have a greenhouse unit at CIRAD, and we have decided to work with different concentration of biochar. And here we have some picture of the preparation of the different pots, and we started with germination, which are here, and here we have the development of the plant, and in this case the plant is sorghum. because we have decided to that. So here you have some information about the agronomic trial which is in progress. So we have decided to work with a model plant, which is a sorghum. Why sorghum? Because it's very interesting in terms of food and not food use, and also it's a plant which is very adapted to water restriction. So it's very, in the idea of climate change, it's very interesting to work with a plant which is very flexible in terms of water adaptation. We decided to work with two biochar, different biochar, which are the risers at 800 degrees C under nitrogen and oak wood at 800 degrees C. And we produce it, as you can see during the presentation. And we decided to work with the different conditions. So the two biochar, one control trial with the soil, that means without biochar. We decided to work with two different soils, one acid, another one alkaline, and three different biochar concentration, 2 tons per hectare, 5 tons per hectare, and 10 tons per hectare. And for each one, 5 repetition trial. And here you can see the preparation of the soil with the mixture of nutrient soil here, nutrient here, and the biochar. In this case is the rice husk biochar, and in this case is the oak wood biochar. So we mixture the preparation in the different concentration, and after that we introduce it into each pot, and all measurement and all preparation is done with a balance, very detailed and precise measurement. So that's all for my presentation. I was very happy to present some results, and if you have any question, don't hesitate. Thank you very much.